Right. So let's talk about something different here. Let's talk about how we can analyze transcriptional regulatory sequences. Now, we talked about DNA, regulatory pieces of DNA, like promoter regions, for example, or enhancers. These are the binding sites for specific promoters, specific regulatory proteins. General transcription factors, specific transcription factors, like um, cyclic MP response element binding protein, for example. It binds to a specific promoter proximal element. There are specific proteins that bind to enhancers. Okay, these proteins cannot bind to other enhancers. Well, so let's say that I want to study how good a gene is expressed in a certain cell type at certain conditions. I don't know, in the presence of hormone versus absence of this hormone, let's say. Um, in the presence of light versus in the dark, and so on. So we want to study how good, how efficient a promoter is at certain conditions in expressing a certain genes. Now, note, I am not interested in studying the gene itself. I'm interested in studying the promoter of that gene, not the gene itself. Okay, but I want to study how good a promoter is at certain conditions. So we can use DNA cloning, the creation of recombinant DNA, to do so. So what are the different transcriptional regulatory sequences? Well, here we're talking about the promoter, we're talking about the promoter proximal elements, we're talking about enhancers and silencers. So we have a gene, we have transcription starting point right here, right? And right before the transcription start site, we have the different regulatory sequences. You have the promoter, promoter proximal elements, enhancers, and so on. Now we want to study this, let's say we want to study this region right here. Now note that we can have silencers instead of enhancers, okay? So let's talk about something different now and see how we can combine all this knowledge together. So we take advantage of an enzyme that is expressed in fruit flies. Okay, so fruit flies are these insects, and their butts glow, right? They light up, creating this beautiful scene at night. And the enzyme is called luciferase. Luciferase. Okay, and what it does is that there is a substrate called luciferin, and this luciferin is cleaved or converted by the luciferase enzyme into oxyluciferin, and this is the molecule that lights up. So I don't care about the reaction itself. All I care about is that we have this enzyme and it cleaves luciferin, creating a product that lights up. Okay, so how can we take advantage of all these tools, molecular tools? So that's what we do. Okay, we create recombinant DNA using these plasmids. So we have a plasmid, bacterial plasmid, and we take the fruit fly luciferase gene and we add a promoter that we want to study. So we're not using the promoter, the original promoter of the luciferase gene, right? So we have it under control of a promoter of interest. So let's say that we have the luciferase gene be under control of the enzyme, the hydroxylase enzyme, the dopamine hydroxylase enzyme, for example, the promoter of the dopamine hydroxylase gene. Okay, and so we create this recombinant DNA, and we transfect, we insert the plasmid into a human cell, a nerve cell, let's say. Okay, so now this nerve cell has its own DNA of interest. Okay, now we can place these cells under different conditions. Okay, and we can then study the expression of the luciferase gene, which is under the control of the promoter of interest, under these conditions. In the presence of light versus dark, in the presence of a hormone, in the presence of a certain neurotransmitter, stress, you name it, okay, you name it, it doesn't matter. Now, we can then add luciferin, and luciferin, if luciferase is expressed, it's going to convert luciferin to this molecule that lights up, the product that lights up. Now, the stronger the signal, the stronger the light, the more the gene is expressed, or let me say, the more the gene is, the higher the activity of the promoter is, the more the gene is expressed. The more luciferase is in the cell, the more light is gonna be produced. Okay, so that's the idea. So we place these cells in different conditions. In the presence of hormone versus absence, neurotransmitter, yes, no, stress, yes, no, and so on. And we measure the amount of light. And that gives us a signal, an idea of how this promoter is regulated at different conditions. Okay, that's one thing. Something else is that we want to study this regulatory sequence. Totally different idea now, okay? We want to study what we know that this region contains regulatory sequences. Promoter, proximal element, promoter, enhancer, whatever. I want to study the position, the location of all of these regulatory sequences. The exact position of the core promoter, the exact position of an enhancer, the exact nucleotides that control each one of these. So we do this. So we have now the luciferase gene, which is right here, the luciferase gene under control of a promoter of interest. And I don't know what the different sequences represent. Now I want to say, here we have a repressor region, here we have an activating region, and here we have the core promoter sequence. Now I don't know what the positions of these sequences are, and that's what I want to study. So I take the DNA piece that contains what I think is the promoter region containing all of these regulatory sequences. So I take it, I clone it into a plasmid. I clone it into the plasmid that contains the luciferase gene. I create a recombinant DNA here. And I take this recombinant DNA, this plasmid that contains the luciferase gene that is under control of the promoter that I want to study. Okay? And I insert it or transfect it into certain cells, nerve cells, skin cells, stomach cells, whatever. And I study how the luciferase gene is controlled by these sequences. And what I do is the following. So I take the luciferase gene the recombinant DNA that contains the luciferase gene, and I place the promoter that I want to study. Okay, the complete promoter. Okay, this whole thing that contains the core promoter, the activating region, the repressing region or repressor region. And I start creating different fragments of this right here. So I start chopping up, I start deleting different regions. So for example, I remove the part of this region, I remove this region, I remove this whole thing, I remove this thing right here, I remove this core promoter complex, and so on. So I have, again, the luciferase gene under control of the whole promoter region, the whole thing right here. And then I start chopping up this piece of DNA. So I remove this part right here. Then I remove this part, creating this. I remove this part right here. So I have now the luciferase gene under control of the core promoter and the activating region. And I remove this part, I remove that part, I remove this whole thing, and so on. Now here I have a piece of DNA that contains no promoter whatsoever, just that luciferase gene. Now the luciferase gene is right here. So this piece of DNA right here is represented by the black arrow. Okay, now, so let's look at the results. Now I transfect, so I have different plasmids, different promoter, pieces of promoter, the same luciferase gene, nothing is changed with the luciferase gene, and I transfect these different plasmids into cells. So each group of cells would have a recombinant plasmid DNA containing the luciferase gene and different pieces of the promoter. Okay, and I measure the amount of light that is produced by converting the luciferin substrate into the light product. Okay, great. Now, I have to have controls. I have to have things that either work or do not work, so that I can compare my experimental DNA with the controls to make sure that the conditions of my experiment are fine. So I create negative controls and I create positive controls. That is, something negative control is something that I know does not work. A positive control is something I know works for good. Now, if the negative control works and it produces luciferase, it means that there's something wrong. If the positive control that should work, if it doesn't work, it means that again there is something wrong with the DNA that I've prepared. Okay, so here we have the luciferase gene with no promoter whatsoever. I know that the luciferase gene does not have a promoter. That's a negative control, something that I know does not work. And here we have the luciferase gene that is under control of a good promoter, a different promoter, a promoter that I know works great. So that's a positive control, just to make sure that the conditions of my experiment are fine. So, and that's the amount of light right here. Okay, so here we have the luciferase gene under control of no promoter. There is very little signal. So someone would say, whoa, wait a second. There is no promoter. Why is there a signal? Well, remember when I talked about leakage of promoter regions? Genes can be expressed even though they have no promoter, even though there are no activators, and so on. Well, there's little signal. So that's a negative control. That's what no expression means. Well, there is some expression, but that's what no expression means, because now the luciferase gene is not under control of any promoter. Okay, here we have luciferase gene under a good promoter, different promoter again. That's a positive control. There is excellent signal right here. Again, there's a lot of expression, 100%. So that's my 100%. Okay, now here we have the luciferase gene under control of the promoter that I'm testing under certain conditions, the whole promoter. Okay, here we have a signal. That's what an expressed luciferase indicates when under control of this promoter under study. Okay, so we have this signal right here. Okay, then what I start to do is I start removing pieces of DNA, pieces of the promoter region. So I start removing the repressor region. So what do you think would happen? If I remove the repressor region, I should see an increase in expression because now there are no repressors that can bind to the promoter and can inhibit the expression of the luciferase gene. Okay, so by removing the repressor region, I should get increase in expression. And that's exactly what happens. So when under control of the whole promoter, including the repressor region, I get this amount of expression. If I remove the repressor region piece by piece, little by little, I get more expression. Wonderful. So I know that this region contains a repressing region. Okay, now let's say that I start removing, now I remove this whole thing, right? This is what I get. Now, there's basically, by the way, no difference between this and that. Okay, so this is just experimental conditions. Now, I start removing the activating region. What do you think would happen if I remove the activating region? I should see a reduction in expression, and that's exactly what I see. If I remove the activating region, there is reduction in expression of luciferase again. Okay, there is reduction in the amount of light that is produced. Okay, so and that's exactly what we see. There is complete reduction in expression of the luciferase gene by removing the activating region. So I know that this region contains an activating region because when we remove it, there is no expression of the luciferase gene. What would happen if I remove the core promoter? Even more reduction of expression of luciferase, less production of light, and that's exactly what I see. This here is similar to this right here, right? Because that's what no production of luciferase means. Very little production of light. Same thing here. Okay, so by removing the core promoter, there is no expression whatsoever. So this is called the luciferase reporter gene assay. The luciferase reporter gene assay. Assay means experiment that measures something. Reporter gene is a gene that gives us an idea. It reports something. And this gene is the luciferase gene, the fruit fly gene, that converts luciferin into a product that produces light. The more luciferase we have, the more light we have. So here we have an experiment where we want to identify the function of different promoter regions, different pieces of a whole promoter, whether this sequence represses, inhibits protein expression, or activates protein expression, or if it is essential for gene expression, that is the core promoter. Or we can place it inside cells, we can transfect cells, and we can put these cells under different conditions again, in the presence of hormone or no, stress or no, nutrition or no, you name it. Okay, and we can measure how a promoter, a specific promoter, functions, how it behaves under different conditions by measuring the amount of luciferase that is produced, which is represented by the amount of light that is produced as well. Allah ma'akum. Now, let's go into something similar but different at the same time. How can we understand protein-protein interaction? Now, I'm going to introduce two techniques to you. One of them is known as co-immunoprecipitation. Precipitation means, it settles down. بترسب, ترسيب. Immuno, once you hear the word immuno, it means I'm using antibodies here. Co means together. Okay, so I'm precipitating a protein with another protein that is accompanied by another protein using antibodies. Remember that we can have, let's say, a large protein. Let's say this protein MLL has two domains, an N-terminal domain and a C-terminal domain, and the N-terminal domain can interact with these two different proteins. And these two proteins can also interact with each other. Now, the C-terminal domain can interact with 4 proteins. Some of them can interact with each other and others cannot, like this protein right here cannot interact directly with this protein, and so on. Well, how can I identify and discover the protein-protein interaction? So with the immunoprecipitation, that's what we do. We have these protein complexes. A protein complex is when you have multiple proteins interacting with each other. What we do is that we add an antibody, and this antibody is specific to one single protein. Now, this antibody then targets this protein, the pink protein right here, and the pink protein interacts with other proteins. So what's going to happen is that this is really quite heavy, and it's going to precipitate. It's going to go down. And we remove everything else, everything that does not interact with the antibody would be removed. And then we have these proteins right here, or the protein complexes, I'm sorry, that interact with the pink protein. We can take this complex and we can study it using, for example, STS page, gel electrophoresis, or using Western blots. Remember, Western blots are immunoblots, where we separate proteins based on size, and then we transfer it to a membrane, and then we add a primary antibody and a secondary antibody to detect the presence of a protein, and we can also determine the size of that protein. Okay, so you can go back to the biochemistry lectures again. Okay, so now by precipitating, by isolating this pink protein, we can identify all of the other proteins that can interact with that pink protein using, again, immunoblotting. That's immunoprecipitation. Let's go into something a bit more sophisticated. Okay, it's known as the yeast to hybrid system. So it's a genetic system where we express hybrids of 2 proteins or 2 genes. So first, I'll give you an introduction. There is a genetic system. There is sort of like a regulatory sequence called UAS. Okay, it's called UAS. This is like a promoter proximal element, you name it. It's the binding site of a transcription factor. This transcription factor is known as the GAL4. GAL4. This GAL4 protein has two domains, a DNA binding domain and an activation domain. Okay, let's go back to the definition of a domain. A domain is a three-dimensional structure of a protein, and it can fold independently of the rest of the protein, and it maintains its function. So we have two domains, DNA binding domain and an activation domain. This activation domain can activate the expression of a gene. Except that the gene that we use, see, that's the power of genetic engineering. The gene that we use is a reporter gene, just like luciferase. Luciferase is a reporter gene. It's a gene that gives us a piece of information. It reports something. It's a reporter. The gene that we use for the yeast to hybrid system is the LAC-Z gene. It's the beta-galactosidase gene from bacteria. It's the gene that produces galactosidase, which is an enzyme that cleaves lactose. Okay, so we use a regulatory element from yeast, and we put it in a DNA upstream of a reporter gene, and the reporter gene is the bacterial beta-galactosidase gene that cleaves lactose. Now, okay, got it so far? So, if LAC-Z is expressed, if beta-galactosidase protein is produced, then it can cleave lactose, except that we use a substrate that is similar to lactose, and it's called X-gal. So it looks like lactose, except that beta-galactosidase would cleave X-gal, producing this molecule right here, which gives a blue color. So the yeast cells become bluish. Okay, got it? So we have this genetic system. Again, the reporter gene is now under control of the UAS genetic system. The UAS system or element is the binding site of the GAL4 transcription factor, which has a DNA binding domain and an activation domain. If GAL4 binds to UAS, then we have expression of the reporter gene by the RNA polymerase, and then translation of the messenger RNA into beta-galactosidase. If we add X-gal to these yeast cells can take up X-gal, they can cleave it, and they can produce this substrate that gives a blue color. Now remember that if we grow yeast on a plate, on a dish, each cell can form a colony. So right here we have different colonies. Some of them are white and others are blue. The bluish colonies are the yeast cells that express the reporter gene. And the white yeast colonies originate from a yeast cell that does not express the beta-galactosidase. Got it so far? Okay, so let's, can we zoom in? No, we cannot zoom in. Okay, so that's the idea. So we have these white colonies that do not express LAC-Z gene or do not produce the beta-galactosidase, and we have the blue colonies that produce the beta-galactosidase. Okay, so what do we do? Remember what the definition of a domain is. So we create a recombinant protein. And the recombinant protein is the following. So we have gene A that has two domains, and we have gene B that has two domains. Well, we can create a recombinant gene that has the portion that produces this domain from gene A and this domain from gene B, and we can put them together. So we produce a recombinant protein that has two different domains from two different proteins. Remember that this domain maintains its function, and this domain maintains its function as well. So remember that. So how can we, what's the purpose of the yeast to hybrid system? To test if protein A can interact with protein B. How do we do that? Well, we use the genetic system that I just told you about, the UAS with the GAL4, which has two domains, and we have the reporter gene, the LAGZ, and we add to the yeast cells the X-GAL. So what do we do? Now, we want to test if protein X interacts with protein Y. We're going to use the same genetic system that we just talked about. The UAS regulatory element, GAL4, and the LAGZ gene that produces the beta-galactosidase. And that's what we do. We create two plasmids. And these two plasmids, they are expression plasmids. We're going to put on one of them gene X, the gene for protein X. And it would be produced as a recombinant protein with the GAL4 DNA binding domain. So when X is produced, it's produced with the GAL4 DNA binding domain. And we have plasmid 2. And this second plasmid contains the gene for protein Y. And it's produced as a recombinant protein along with the GAL4 activation domain. So we're going to have protein Y with the activation domain of GAL4. The idea is that if X interacts with Y, then these two would be close to each other. So we're going to have, again, this protein of interest with the DNA binding domain. And we're going to have this another protein of interest, protein Y, with the activation domain. Now, if these two interact with each other, then we're going to have the DNA binding domain and the activation domain very close to each other, and we're going to have transcription of LAGZ. If LAGZ is expressed, we're going to have beta-galactosidase. If beta-galactosidase is expressed, if it's there, it's going to cleave X-GAL. If X-GAL is produced, is cleaved, it's going to generate a blue product. And the yeast cell and the yeast colony becomes bluish. And the color blue indicates that X interacts with Y. If X does not interact with Y, then the DNA binding domain would not be close to the activation domain, and as a result, there would be no transcription of the LAGZ gene, there would be no production of beta-galactosidase, there would be no cleavage of X-GAL, there would be no blue product, and the colony would look whitish, not bluish. Got it? So we have these two probabilities. Either X interacts with Y, a blue colony, X does not interact with Y, white colony. White colony. So the previous example was simple. I'm dealing with X and Y, just one single protein, X, and one single protein, Y. Now, let's make things a bit harder. I want to identify all of the proteins that can interact with X. Not a single protein. I'm not testing a single protein. I want to identify all of the proteins that can interact with X. So, we create a cDNA library. Remember what it means? It's a collection of plasmids, and each plasmid contains a cDNA representing a specific gene. That's a cDNA library, a collection of different plasmids. So I have plasmid, this plasmid right here producing protein A. It contains the gene for protein A. And this plasmid has gene B for protein B. This is protein C. This produces protein C. This produces protein D, and so on. This is a cDNA library. So I have different plasmids that can produce proteins, recombinant proteins. Each protein is produced with the activation domain of Gal4. And I have one single plasmid that can produce protein X, and this protein X is produced as a recombinant protein with the DNA binding domain of Gal4. So we take this and we introduce it into yeast cells. So this plasmid right here that produces recombinant X would exist in every yeast cell. And I have, let's say, 1 million yeast cells. Each one of them has the plasmid for the recombinant X gene, or that can produce a recombinant X protein with the DNA binding domain. And then I take these 1 million yeast cells and I insert these plasmids into these yeast cells. So each yeast cell would take up one of these plasmids. Okay? So each yeast cell would have two plasmids. The plasmid that produces protein X, recombinant protein X, and a plasmid that would produce either recombinant protein A, or recombinant protein B, or recombinant protein C, or recombinant protein D. Okay? So each yeast cell would have two plasmids. Now, and we grow them on a plate. If X interacts with A, then this yeast cell would have the production of the beta-galactosidase that can cleave X-gal into a bluish product, and the colony would look blue. If in this yeast cell X does not interact with protein B, it means that the binding domain and the activation domain would be far away from each other. They wouldn't interact with each other. They wouldn't be close to each other. And as a result, there would be no production of beta-galactosidase. There would be no cleavage of X-gal into a blue product, and the colony would look white. So we can have a plate that has a lot of yeast colonies. Some are white, some are blue. And I'm interested in the blue colonies. Okay, so what we do is we take these blue colonies and we isolate the plasmids, and we see what the identity of the gene that is contained in these plasmids. Now, each yeast cell or colony would have one of these plasmids. So what we do is that we take these plasmids, okay, we grow the yeast cells, we isolate the plasmids, and we see what the identity of the gene that is included, that is cloned into this plasmid is. And we can identify what the protein is, or what the gene is. And this is the gene that can produce a protein that interacts with X. so we can identify the X partner. All right. So these are the probabilities now. If we have a plasmid that produces the full Gal4 protein, this is the binding domain, the binding domain, and the activation domain, then LAC-Z would be expressed. Beta-galactosidase would be produced. And if we add X-gal to these yeast cells, we're going to have a blue colony, right? Because we have the whole Gal4 protein. If we have a plasmid that produces the DNA binding domain only, then there would be no production. If we have a plasmid that produces the activation domain only, we're going to have no production of beta-galactosidase, okay, because we need the DNA binding domain in here. But if we produce protein X, protein Y, and these two proteins can interact with each other with the DNA binding domain being recombined, being part of protein X, and the activation domain being part of protein Y, then these two would be close to each other. The activation domain, the DNA binding domain, we're going to have expression of LAC-Z, production of beta-galactosidase, conversion of X-gal into a blue product, and the colony would look blue. We take these cells, this colony, we isolate the plasmid that contains this gene, because I know protein X or gene X. I want to identify this gene or protein that can interact with protein X. So we isolate the plasmid, and we can do sequencing, we can do immunoblots, whatever, and we can identify this unknown gene. Okay. So again, I'm interested in the colonies that are blue. These are the colonies where we have yeast cells that produce a protein that can interact with protein X. So I'm going to leave you with this slide. This is taken from the book. It contains a lot of information. Okay, it explains the technique that I just told you about, and you can read it on your own. Okay, and Allah ma'akum.